Hello everyone, welcome to sqadia.com. Today we are at the discussion of cellular energetics, and after discussion the glucose metabolism, after discussion of the fatty acid metabolism and the citric acid cycle, we are at the control of metabolism. And in this section we are going to start off with the discussion of the fatty acid oxidation in mitochondria and the oxidation of the fatty acid in the peroxisome. That would be a comparative figure that is going to explain minor differences between the oxidation of the fatty acid that occurs in mitochondria versus peroxisomes. Then we are going to study the allosteric control of glucose metabolism, of how metabolism is being controlled by primarily by an enzyme which is being activated by 2-3 different factors that plays an important role, which are activated or inactivated in the absence or in the presence of glucose. So this would be followed by the discussion of changes in the redox potential and changes in the free energy, which is, in this case, we are basically going to understand how when the electrons are being donated from NADH to the electron transport chain, these electrons are going to move down their energy gradient, losing their energy, and this energy is being taken up is utilized by the protons to move against their concentration gradient to establish strong gradient down stream, so that they can move in the presence of ATP synthase down their concentration gradient, resulting in the production of ATP that we are going to discuss in the next section of the generation of proton motive force. So we have a fatty acyl-CoA that has been produced from the fatty acid. Now fatty acyl-CoA is going to get the action of the dehydrogenase, and of course it would be produced into the fatty acyl-CoA. This of course would produce the FADH2 molecules. Now if you recall, we are going to study in detail the FADH2 molecules as well and the NADH2 molecules as well. These FADH2 are going to move into the respiratory chain, and when they are going to move into the respiratory chain, they are going to be converted into FAD, phenylbenzene deoxyribonucleotide, resulting in the production of ATP. And this would, of course, because the two hydrogen molecules are going to be catered with the help of the oxygen molecules and would be converted into the water molecule as well. Comparatively, in case of the peroxisome, this FADH2 that is being produced would undergo oxidation by an enzyme oxidase and would be converted back into FAD, and this oxidase would help in the production of from the oxygen to H2O2, which would be a highly reactive intermediate and would be converted down in the presence of the catalyst back into H2O and water and oxygen molecule. So this intermediary step would undergo the further modification in the presence of the addition of water by enzyme hydrolase and would be converted into fatty acyl-CoA. So fatty or the acetyl-CoA. Now what happens in this case? The acyl-CoA, which is being produced, would further undergo the dehydrogenation. Now dehydrogenation means that the hydrogen is being removed. Now this removal of the oxygen by the dehydrogenase, after it has been removed, this hydrogen is going to be given to NAD positive, which is going to be converted into NADH, that we have already discussed in the previous section of how the whole process would generate an FADH2 molecule and NADH molecule as well, which both of these would go to the respiratory chain or electron transport chain, resulting in the production of course, ATP through the ATP synthase. So what happens? This would be followed by the action of the thiolase enzyme, where coenzyme A is going to occur, is going to act on it, and this would be converted into acetyl-CoA, where acetyl-CoA is going to cater only 2 carbons. So acetyl-CoA, in case of the mitochondrial oxidation, is going to enter into the citric acid cycle. In case of the peroxisome oxidation, acetyl-CoA is going to be exported out of the peroxisomes. So moving towards the allosteric control of glucose metabolism, we have phosphofructokinase 1 this enzyme that plays a critical function in the allosteric control of the glucose metabolism. Now, this phosphofructokinase is going to be positively regulated by fructose 2,6-bisphosphate, which is produced when the insulin levels are high. So, in the presence of insulin, it means that the glucose levels are low. The high level of the insulin is going to favor the conversion of fructose 6-phosphate to fructose 2,6-bisphosphate with the utilization of a molecule of ATP. So phosphofructokinase 1 and 2 both plays an important function, and it's important to consider the high ATP, since high rate of ATP, or if there is an abundant amount of ATP that is present in a cell, and cell does not need any kind of the ATP generation at that particular moment, the high ATP is going to negatively regulate the phosphofructokinase 1, so that fructose 6-phosphate is not going to be converted into fructose 1,6-bisphosphate. Same goes for the case of the citrate. If there is a high rate of the citrate, it means that the process of the glucose metabolism would hamper at the fructose 6-phosphate conversion to the fructose 1,6-bisphosphate. However, if there is a high level of AMP, it is going to positively regulate, along with the fructose 2,6-bisphosphate, the activity of phosphofructokinase 1, so that the glucose metabolism is going to be preceded, and further it is going to be converted into pyruvate, which is going to be converted into acetyl-CoA, and this acetyl-CoA, of course, would enter into the citric acid cycle, yielding NADH molecules, FADH2 molecules, carbon dioxide, and these NADH and FADH2 molecules would then enter the respiratory chain, where they are going to ultimately yield ATP molecules. So, talking about the changes in the redox potential and the free energy, we have over here the you can say the electron transport chain or the respiratory chain. So we have NADH that has reached the initiation of of course, the respiratory chain. So this particular complex, we have two carrier transporter molecules, the CoQ and the cytochrome C, that basically helps in the movement of the electrons from one complex to the two complex. So we have four complexes of FMN, or the flavin mononucleotide complex, the FAD complex, the cytochrome complex, and of course the cytochrome oxidase complex 3 and so on. Now over here, this is CoQHQ cytochrome C reductase complex, which is to simplify is known as complex 3. This is complex 4, complex 2, and complex 1, of course. So what happens, an ADH is going to be converted because it is going to give 2 electrons to FMN, and these 2 electrons are basically, if you can see, the redox potential moving down is increased from upward to inward to downward, and this free energy that these have are going to be decreased slowly and steadily as they move down the redox potential. So as they are moving from the around 300 millivolts to around 150 millivolts, this energy is being utilized to move the protons from inside to outside. So similarly, these protons are then going to get entered into the CoQ, where it is going to play an important function in the formation of FADH molecules in the complex 2, as we saw as well. So moving on, after the FADH2 have been produced, the molecules are going to move down this CoQHQ cytochrome C complex, cytochrome C reductase complex, of course, which is simplificationally known as complex 3. This decrease in the redox potential and free energy would additionally will cause the movement of the protons from inside to the outside against the concentration gradient. It's important to consider the protons are moving against the concentration gradient. So when moving towards the cytochrome C oxidase complex, what happens? These electrons are passed from the plastoquanone plastocytine complex as well, and that's not, of course, shown over here. The cytochrome A, B complexes as well, A3 complexes as well, and then they are moving down where the redox potential is being increased continuously and their free energy is being reduced. This complex would also allow the further movement of proton from inside to outside against their concentration gradient. and ultimately these electrons are being given to the molecules of oxygen that are being produced along with the protons, and a molecule of water is being produced. So to summarize this particular slide, we had NADH molecule. Two electrons of the NADH allowed the movement of protons from inside to outside and yielded a molecule of water as well. So in this section, we covered the fatty acid oxidation in mitochondria and peroxisomes. We discussed the allosteric control of the glucose metabolism, changes in the redox potential and free energy, and how we saw the movement of the electrons down their energy, free energy, where losing their free energy is being utilized by the protons that are present inside to move against their concentration gradient to move outside. And that was all for this section. I'll see you in the next section. Keep watching sqadia.com.